Hello everyone. Today we will be solving a question which came from the topic of molecular biology techniques, and it came in the year 2015, and the question number is 20. The question is stating is given below are the maps of a 1200 base pairs long DNA insert and a 3000 base pair expression vector. The BAM-H1 and HIN3 restriction sites and DNA length between them are indicated in base pairs. The insert is cloned into the vector at the BAM-H1 site and the desired orientation is shown by the arrow. After cloning, the orientation of the insert in the recombinant plasmid is tested by complete HIN3 digestion followed by agarose gel electrophoresis. Which one of the following band patterns reveal the correct orientation of the insert? So it is something which is related to the topic of molecular cloning and how these restriction enzymes are actually working, and also you have to know how this DNA actually migrate in a agarose gel electrophoresis. So in the next slide, they have given actually four options that how after digesting your enzymes, your construct with these enzymes, how many bands you actually will find in your sample. So for knowing into this answer, first we need to know something about the molecular cloning. So what happens in the molecular cloning? In molecular cloning, what happens is that the source of interest with which you start to clone, that from which you will actually the starting point from where you will get your target sequence, that what sequence to clone, that can be your total genomic DNA or that can be your cDNA. So why cDNA? Because if it's a eukaryotic protein, then if the main purpose of this cloning is to express our protein from the constructs that we are actually cloning. Okay, so for expressing our proteins, we need a sequence which will contain all the exonic sequence only, because in eukaryotes there is this post-transcriptional modification that can happen, but in normally we actually clone these constructs with the help of the prokaryotes like bacteria, like the E. coli bacteria strain, different strains of E. coli like DH5 alpha strain. Okay, so there it does not contain this post-transcriptional modification. So if we start with a genomic DNA, so the problem will be, and if you are actually working with a eukaryotic protein, then the problem will be there will be some intronic sequence also present in your construct. So to avoid that, what we do is that we isolate the total RNA, from that total RNA, we need to actually take out the mRNA, and from the mRNA, we convert this mRNA to the cDNA and use it as our source of interest. This conversion is being facilitated by the reverse transcriptase enzyme. Then what we do is that we have a vector which will actually contain three basic sites that it should contain. Okay, so one is this oricenter from where the origin of replication will start. Then it will have a selectable marker which generally consists of selectable marker antibiotic resistance genes. Okay, antibiotic resistance genes will be present, and another site where it will have a multiple cloning site. So multiple cloning site is the region where we will be actually cloning our construct. So these multiple cloning sites contain large amount of restriction enzyme sites. So what are the restriction enzymes? These are certain enzymes which require palindromic sequences, specific palindromic sequence, and cut in that specific palindromic sequences. Okay, so this BAM-H1 will recognize its specific palindromic sequence, NDE1 will recognize its specific palindromic sequence. So if in the PCR, if we are actually amplifying our construct from this cDNA, we use two primers. And in the flanking sequence of the primers, we have the restriction enzyme sites for two restriction enzymes. For example, here, let us take BAMH1 and ND1. We will cut the vector with the same restriction enzyme BAMH1 and ND1, and the insert which has been amplified from the cDNA using PCR using the same set of restriction enzyme. Then we will be actually ligating it with the help of certain ligase like T4DNA ligase in presence of T4DNA ligase buffer, which it actually requires ATP. And then after the ligation, since two staggered ends will be created, then there will be double digestion. We actually transform it into a E. coli bacteria, and the transformed, if and we grow this bacteria in a selectable marker gene, antibiotic selectable resistance marker gene. In that antibiotic, which actually contains the antibiotic resistance genes which is present in the vector, we will have to know that, and on the basis of that we will be actually growing this bacteria in a plate accordingly. Okay, so if it is containing ampicillin resistance gene, only those constructs which have actually been successfully cloned and being transformed in the bacteria, only those bacteria will be able to survive in an ampicillin plate, agar plate. Okay, so in that way we will be able to screen the colonies. Okay, so this is in a nutshell what is about cloning. So after the cloning, as I said, that the main goal of our application is that we actually express those proteins, and after the expression, we actually perform the purification of the protein. I am not going deeper into each of these steps. So now let us look back into our question and try to solve it. Okay, so first, what is being done here is that the total length of the vector is being given. Given below are the maps of your vector, that is then the insert, and the 3000 base pair extension vector. So the vector length is given as 3000 base pair, and the total insert length is given as your 1200 base pair. And it has been said that there are two restriction enzyme sites, that is BAMH1 and HIN3 sites, and the DNA length between them is indicated in base pairs. So in each of these sites, how much length difference is there, that has been provided. The insert is cloned into the vector at the BAMH1 site. So at the BAMH1 site, so in both of the insert and the vector, it has been cut with BAMH1, and the insert has been actually cloned into the vector, the BAMH1 site, and the desired orientation is shown by the arrow. So there is a desired orientation, means if you put your insert, if you cut here with BAMH1, it will be giving a linearized fragment with two BAMH1 side. You need your insert to be put in this direction. So if you see 5' to 3', so 5' to 3' should go in this direction in which the arrow is pointing. Okay, so after cloning, the orientation of the insert in the recombinant plasmid is tested by complete HIN3 digestion. So this orientation, whether it is in the correct orientation or in a reverse orientation, that is being actually tested by a complete digestion by HIN3 and then running it to gel electrophoresis. So which of the following band patterns will reveal the correct orientation of the insert? So if it is correctly oriented, then what will be the band pattern of your insert if you run it in a agarose gel electrophoresis? So this is the question. Now let us look into the answer and solve it step by step. On cloning the insert in the vector, the recombinant vector formed would be. So what will be the recombinant vector will be looking after cloning this insert into the vector? So you cut with BAMH1. So this will be the orientation. So BAMH1, HIN3, BAMH1. Okay. And I have calculated the total. The total length is given as 3000, and all these lengths are provided. So accordingly, this length from BAMH1 to HIN3 will be 2700 base pair. It has 3 HIN3 sites now. Okay, after the cloning has occurred. On complete digestion by HIN3, the products possible are. So if it is completely digested, then all the 3 HIN3 will be cut. but there will be possibility that it is partially digested. If when there is a cut only at the single hindry side, if there is only cut in single three hindry side, so there can be three possibilities. One in this hindry side it can cut, one in this hindry side and one in this hindry side. So three possibilities can be there. So I have put all these three possibilities how it will look like. Okay, but the whole length of your plasmid will be 4200 base pairs only. If you add all these numbers, you will get up to 4200 base pairs. Okay. Now if you cut on two hindry sides, so either this one, or this one, or this one. Okay, so there can be different possibilities. So again, all the possibilities I have actually drawn, that how it will look like if there is cut in two hindry base pair side, and if it is a complete digestion, that all the three are being cut, then your total length will be 3000 base pairs. One will be 3000 base pair, one will be 600 base pair, and one will be around 200 base pair. So now just there is also a molecular weight ladder marker which is given. You just need to compare this length with this gel image. So you see in the option A, you have a band around 3400. You again have a band around 600 base pair, and a band around below this 250 mark, that is around 200 base pair. So the answer of your question will be option A. For your reference, you can actually look back into these two courses of NPTEL. So the first course is of Experimental Biotechnology by Professor Vishal Trivedi of IIT Guwahati, and the second course is of Genetic Engineering Theory and Application, again by Professor Vishal Trivedi of IIT Guwahati. So you can look into these two courses for clear your concepts in molecular biology techniques.